In this video, I'm going to take you through how to interpret Rna-Seq data on the Ucsc Genome browser. And I've decided to use this example of the human cyclin-dependent kinase inhibitor 2 genes because it's easy to see different isoforms of expression on this gene, the cyclin-dependent kinase 2A gene, which makes at least two different proteins. The CDKN2A gene provides instructions for making several proteins. The most well studied are the P16-INK4A protein and the P14-ARF protein. Note that the P14 protein is called P19 in other mammals. Both function as tumour suppressors, which means that they keep cells from growing and dividing too rapidly or in an uncontrolled way. Both proteins are also involved in stopping cell division in older cells, known as senescence. Note that the nearby gene CDKN2B makes the INK4B protein, and INK4B and 4A, as you can tell from the name, are members of the INK4 family of proteins, which are cell cycle inhibitors. When they bind to the cyclin-dependent kinases CDK4 and 6, they induce an allosteric change that leads to the formation of CDK-INK4 complexes, and that prevents the CDKs from interacting with the cyclins. This in turn leads to a prevention of these CDK cyclin complexes acting on proteins such as retinoblastoma, or RB here, and preventing it from being phosphorylated. RB, when it's unphosphorylated, is a powerful repressor of transcription of S-phase genes. So if RB is not phosphorylated, then you get an inhibition of S-phase and a cell cycle arrest at G1. The P14-ARF protein protects a different protein. It protects P53 from being broken down. The P53 protein is an important tumour suppressor that's essential for regulating cell division, senescence, and self-destruction, or apoptosis. By protecting P53, P14-ARF also helps prevent tumour formation. The P14-ARF and the P53 proteins are often made in cells that are unable to undergo cell division. So on this Ucsc Genome browser session here, I've started up with HD19 human genome build, and I've got the CDKN2B and 2A genes here. I've reversed the orientation of the browser so they're in the order that you've seen in the previous figure. I've also used the BLAT alignment tool here to take the protein sequences of P15-INK4B, P19-ARF or P14-ARF, and P16-INK4A, and I've aligned those to get an annotation so you can see where each protein comes from these different genes. So you can see that the CDKN2A gene has got two main expression forms, and they make these two very different proteins. If you right click on an annotation track, you can change its different view settings. So if I right click on this here and go to configure genes, I just want to point out that if I indicate splice variants and say OK, then you can see that there are many other recognized forms of transcripts that come from these genes. But for simplicity, I've just decided to hide those for now, and I shall come back to that. Then you've got all sorts of sources of information that's embedded within the browser, or you can easily link to in the browser through track hubs to understand the kinds of Rna-Seq data that you might see for these genes in this case, but Of course, you could be looking at any genes relevant to your studies. So firstly, this project here, GTEx, has done RNA-Seq in lots of cell types, and if you turn that track on, then you can get these histograms that give you a quick overview of cell type specific expression, tissue specific expression. So in some cases that can be very useful for you getting an overview of what kind of genes are next to each other. That's not really the purpose of this talk. In this talk I really want to focus on RNA-Seq data and what it might look like and how you would interpret it. So firstly, just looking at this gene annotation track again, in case you're unfamiliar with it, you can see we've got all sorts of different marks here. We've got thin boxes, thicker boxes, and then thin lines with chevrons on them. So clearly they're all connected together, so they relate to one annotated, well understood transcript. And the chevron indicates the direction of transcription. So the promoter is here and reads through to the end here. And if we look at the beginning and the end, then with many transcripts you'll see these untranslated regions, UTRs. So you have a 5' UTR, which is the thin box, and the 3' UTR, which is a thin box. And sometimes the UTR is split over 2 exons. And then we've got the coding exons, which are the thicker bars. So if I just hover over it, we've got exon 1, exon 2, exon 3. Okay, so RNA-Seq is taking messenger RNA, fragmenting that, sequencing that, aligning it to the genome, and then you can just get these raw tracks of where those fragments, those sequence fragments, align in the genome. So there's an important distinction from understanding transcripts. So to understand alternative splicing, you really do need to look at the sequence and see how specific exons splice onto one another, and from that data you can say what the alternative splicing is of, or alternative isoform expression is of a given gene. So you do that bioinformatically. However, you can also use the genome browser to visually see differences in isoform expression, which can be very useful when you're overlaying with other information such as transcription factor binding, histone modification, and so on, when you're trying to understand the regulation. So for example here, I've identified some RNA-Seq tracks that are built into the genome browser from the Burge laboratory, done quite some time ago, and they're from 4 different breast cancer cell lines. So these are cell lines. So we've got T47D, BT474, and so on. And what you can see is that the RNA-Seq reads are not the same between each of these cell lines, and we've got a different accumulation over the exons. So remember, you're sequencing messenger RNAs that have already been spliced. You're not sequencing nascent transcripts. So your intronic sequences are lost. So inevitably the reads pile up over the exons. They all align with the exons. So that should make sense. And we can see here in this particular cell line, we've got no transcription over exon 1 here. But we do have transcription over exons 2, 3, and all the way over here on this alternative exon 1. So clearly in this cell line, it's expressing P14ARF or P19ARF, depending on what you want to call it. Whereas if we move to this other cell line, then it's expressing both. So we've got some transcript here, but also we've got transcript at this exon here. Okay, so from this you can get these quite quick overviews of the alternative expression that happens, and also the relative levels of expression. I'm going to show you some other sources of information. as an illustration of the kinds of data that are there, and they may prove to be very useful for you interpreting what is going on. Another important thing to figure out is which transcription start site is being used by a given gene, and this can be insanely complicated. So there is a project called Phantom5, an international consortium, that have used an RNA sequencing technology called cage tag sequencing. And what that does is sequence the very 5' end of nascent transcripts. So you get your very beginnings of your messenger RNAs and other RNAs like enhancer RNAs. And they've done this in a very large number of primary cell types and some cell lines as well, mostly primary tissues. And from that, this project has come up with lots and lots of different transcription start sites for each gene, which have all been validated. So you can see here for CDKN2A, we've got multiple different promoters, and we're interested in the forward strand as we're looking at it here. So it's the blue annotations we're interested in. And we can see here that these transcripts are indeed all starting at promoter 1, the main promoter. But it's important to note that there are these alternative promoters as well that might be used in a given tumor type, for example. So if we look in a couple of breast cancer cells here with the Phantom5 data, so these is this cage tag sequencing, and we can see that there are tags accumulating again at promoter 1 for these particular 2 cell lines, so MCF7 and this one here. So that can be very useful, and there'll be a separate video on how to find this Phantom5 data and set it up and make sense of it. But this can be very, very powerful. And if you're going to do anything where you're trying to inhibit a promoter, for example, you want to use CRISPR inhibition or CRISPR repression, or you want to study a promoter and get to where the regulation is actually occurring, then it's really key to know exactly where the transcription start site is for your given cells, because the annotations here are an average of everything known, and these annotations may not be specific to your given cell type, and that's very important to understand. So this next data set below that I've decided to highlight is from the TCGA project, where they've looked at gene expression in lots of different tumor types, primary tumors, and they've decided to present it as the classic red and green of expression going up and down, and you can very quickly scan through and see whether a given gene is up or down regulated in different cancer types. As we hover here, we can see the different cancers on these little screen tips that are popping up. So that might be particularly useful if you've got a gene that you think is involved in cancer. And then finally, I wanted to highlight an important function of the browser in terms of how it displays data to you. So firstly, I'm just going to drag and drop this gene annotation here. So once you're over a track and it's highlighted in green, and I'm putting my cursor where the labels are. In this case, I've got it on the right hand side. It may be on the left for you when you start. So once you're highlighted in green, left click and hold and drag down, and I'm just keeping hold and moving to the bottom of the screen, moving up again. And then once I've got it where I want to, I'm going to let go of my mouse key, and this track is now next to this other set of Rna-Seq data. And if I highlight here, doesn't say unfortunately. So this is all data from the NIH Roadmap project, where they've done Rna-Seq in lots of primary tissues. It's very nice quality data. And as we scroll through, I'm just going to scroll down for you. We can see there's lots of data from lots of different tissue types, and you can see the RNA-seq profiles are quite different between cell types for these genes, and some of them look quite strange like this, and then some of them look really quite straightforward and very clear. So scroll down a bit faster. Okay, so I want to explain to you what is going on. So firstly, when we look at these consortia data and you don't know anything about a gene and you're just trying to get a feel using the tracks that are available within the genome browser, it's nice to plug into one of these track hubs like this and just turn everything on and visualize it and just compare everything side by side. And what I wanted to point out is if you right click and go to configure, and I'm going to configure the whole set here, so I'm treating all the tracks the same rather than just changing the view of one track at a time, then this is the setup for this particular track hub with all that data. And if I click here, so each particular hub or set of data is sort of arranged slightly different here, but hunt around to see if you've got any settings, and here we've got a common setting, and all of those tracks that you've just seen are all auto scaled to the view that you see, to the window that you're in. So the highest point on a given track is set to 100% of the height of that track. So I'm just going to go back. So you can see here, this is the highest data point for this particular track in this window. If I zoomed out or zoomed in, it would be quite different. So if I zoomed in on this, it would make this peak reach to the top. If I zoomed in here, it would make this flat data zoom up to the top, for example. And then as we scroll down, we've got some tracks which look quite strange, like this one, quite blocky, and all of these here. Well, what's happening is effectively no data. There's hardly any reads on these genes. This is just background read alignment across the genome. It's non-specific alignment. So there's no expression here, but because we've set the browser to say auto scale these tracks to the window, then it's made this background data, these may be just single reads here, of expanded up the full height of the track. So it's very misleading. You cannot compare the different levels of transcript that are expressed in one cell type to another by doing this auto scaling. So what this is good for is, and that track needs to be moved again because it's not remembered it. What this is good for by doing auto scaling is we can get a qualitative view of the data, and we can see which exons are being transcribed and therefore what alternative isoform expression is taking place. By comparing these tracks, you can see how this and this are very different, and these two have a difference as well. So let's now try a new function of the genome browser. So again, I'm going to go to that configure the track set, wait for this to load up, and now we can do something called group auto scale. So I'm going to submit that and see what it does. Okay, so we can see what's happened now is a lot of the data looks like there's no expression anymore. And as we scroll down, we can see there is expression in some cell types. And what it has done, it's auto-scaled to the highest point for these genes in the entire set of data that we're looking at. And it's put all of these RNA-seq tracks on the same normalized scale. So they are now all relative to one another. And because this particular data here. So these are mesenchymal stem cells. Because the CDKN2A expression on this particular exon here is particularly high, that's set at 100%, and everything else has been scaled down to be normalized to that. Okay, so now we can compare the relative expression between cell types, and we can clearly see that these genes are much more highly expressed in these stem cells compared to these other somatic cells above and below. Okay, so that's a very important distinction to make, is being able to auto-scale to a group versus just auto-scaling to one individual track. So I'm just going to show you that again. So now we're auto-scaling to one particular track. The other thing you can do is obviously display tracks in lots of different ways, and if we go on here, so we can go to dense, and instead of getting these full profiles on exons, we can now get these density maps. And again, the same warning comes as before. The shading here, so black obviously means that we've got more reads accumulating on a given exon. Just going to move this annotation again. Actually, I don't know where it has gone. Oh, it's up here now. So as I move this down, we can see that the reads are accumulating over given exons, and the shading relates to how much. But then when we look at tracks with really no transcription, then you get this very confused picture. It's not aligning with the exons. Okay, so that's all about the auto-scaling that's taking place. Okay, so I hope this session was useful to you, and you feel confident about looking at RNA-seq data. Thank you.